confession to make. I'm not going to finish in 15 minutes. So the beauty of being the last speaker of the day is that you all can leave at any time. I won't be offended. Okay. All right, so basically today I want to tell you a little bit about the outcome of two reports that are the product of a collaborative effort between Rancho Santa Ana Botanic Garden and Joshua Tree National Park. Recently we received a little bit of money at the park to fund a small genetic analysis, as well as do some germination trials and write up a report. So basically RSA helped us with those aspects of this. So this map here shows where Joshua Tree National Park is, in case you're not familiar. It's the brown, right here, the brown section there. And Joshua Tree only has two federally listed plant species. That's Astragalus tricarinatus, which is shown in the blue, and Erigeron parishii, which is shown in the red. Here we go. Let's see if I can advance this. So I'm going to try to run through each species individually. Like I said, it's a lot of information, and I had to hack my presentation into halvesies and thirdsies and all kinds of eliminations. So I'll try to get through this as fast as I can. But the beauty of it is you can all go read about this in our report. So if you want to read about it, you can get the report from me or on our website. So Astragalus tricarinatus was listed as endangered in 1998. It lacks designated critical habitat. It is protected under the Coachella Valley Multiple Species Habitat Conservation Plan, which is a mouthful. But this is a regional habitat plan or conservation plan that's just to the south of Joshua Tree National Park. And so obviously it's something that we want to collaborate and coordinate with in the future for this species. As you can see in the picture, it's a very pretty plant. It's fairly distinct looking, I think. It has these cute little gray leaflets that are widely spaced. It has these pale yellow flowers, and then the common name is the triple-ribbed milkvetch, and that's because of these the fruits right here that are three-ribbed. It has this upper suture that has a bit of a ridge on it. So it's an endemic to Southern California. It occurs in Riverside County. This is the county line right here, and just for reference, this is the park boundary again. So Riverside County is where it mostly occurs. It does get into San Bernardino County up here. At the time of listing, there were only 8 extant occurrences, all with fewer than 15 individuals. Currently, we have 46 occurrences. The estimated global population size is around 3,100 individuals from about 10 watersheds. So it's a fairly limited species, both geographically and just sheer numbers. And then I just had to throw this in. I had a separate slide, but figured I'd try to throw this in, combine it with another one. This is a geology map from USGS, very coarse map, but it does show you that the majority of the plants occur on gneiss or granodiorite. There are some occurrences on alluvium. Those, I think, mostly relate to the really small occurrences, which we now consider wave populations. There's a lot of occurrences that have 10 or fewer individuals. So 70% of the known 46 occurrences have 10 or fewer individuals. So again, very limited species. Let's see. And then, so this is a little bit closer look at just in Joshua Tree. Again, the boundary shown in red here. And then the background map is a soil map that was done for Joshua Tree. And this one shows that we do have a lot of mafic and felsic substrates, but again, it's very much granite and gneiss. That's pretty much all we have in Joshua Tree, a lot of granite. And in Joshua Tree, we have 18 known occurrences, 12 of which have 6 or fewer individuals. And in fact, that's these points here all represent these little wave occurrences. And that was in 2006. That's when I found a couple of these, and that was sort of the red flag that, oh boy, I think we probably have some bigger populations. And so we upped our upped our ante and started looking up in these higher reaches of the Little San Bernardinos. And so since 2008, we've added 11 occurrences, which has been a lot of fun. Just to go back over here, so right now we estimate we have 12 acres of occupied habitat with approximately 560 plants in the park. And prior to 2006, we only knew of the species based on 3 historic vouchers, one of which has recently been annotated to Astragalus bernardinus. And then the Eureka peak population, which I'll talk about in a little bit because it's one of the populations we used in the genetic analysis, is right there, and it's now the 3rd largest population for the species, so we estimate around 215 individuals. So in Joshua Tree, the habitat that it occurs on is fairly distinct looking. You can see in this picture it's also very steep and hard to walk on. This distinctive white to gray-green colored substrate is likely due to biotite or biotite hornblende, but we don't have any analyses or anything to show exactly what unique characteristics there are. That is something we'd like to pursue, however. But so in order to look for more populations in the park, we decided to put together a habitat model. And so Sean Murphy was our GIS guru at the park for a while. He's no longer there, unfortunately. But he used Python, which is available online, to create a script, which he then inserted into ArcGIS in order to make a little more user-friendly tool for us to create a habitat model. And it utilizes up to 5 parameters. So for Joshua Tree, we have a lot of information available for the park. So we were able to use, we have the soil map, we have vegetation types, we have slope, aspect, elevation, and then we have a lot of points for where we've seen the individuals. And so what we did is we drew a 15-meter radius buffer around each known location, each individual, and then use those 5 parameters to basically characterize what occupied habitat looks like in Joshua Tree. And then use that to assign probability values across the landscape. And then, so this is the outcome of the habitat model, the first rendition anyway. So we use that to prioritize some ground truthing and searching, and you can see these purple lines here. We even went down here to the Eagles to look for it. Didn't find any there, but did find 2 new populations here, and pretty good sized ones, populations as far as Astragalus tricarnatus is concerned. So we felt pretty good about the outcome of the habitat model. It was very time consuming, so because this first round what we did is we basically were manually entering all of the data, and that was really hard to do. And so before Sean left the park, he actually turned it into an automated script, which in itself has its own biases and problems, but it's still a fairly workable model. And I think it's, as we move on and learn more about the habitat and get more points, then we'll learn more about that, and maybe develop some new versions of it. Okay, so germination trials. So I mentioned that RSA, we contracted with RSA to help us with these projects, and basically they did the germination trials and they also did the genetic analysis and then helped us with compiling all the information and writing up these reports. So their germination trials basically corroborated previous research that's been done that indeed these seeds require some form of scarification to germinate. Clipping the seed coat at the hilum yielded 97 to 100% germination. Scarifying the seed coat using sandpaper yielded 68 to 96, and then there's a slew of other things here. This no treatment, cold water soak, cold moist stratification. All this didn't do very well. So, but this is good information to have for any potential future growing for the plant. The population genetics study was done by Linda Prince at RSA. She used ISSR genotype data from 3 populations. You can barely see the screen from over here though. So there's 2 populations from within Joshua Tree. They're marked with the yellow stars. maybe I'll explain. Yeah. So this is the so population 1 is Eureka Peak. This is the large population that we found, estimated at 215 individuals. This one we're calling the Covington Flats population. We estimate that at 70 individuals, and then one population from outside the park, which is on the Wildlands Conservancy lands. That's at Catclaw Flat. Estimated population size of 100. So she was able to generate 140 polymorphic loci from 3 primers. And indeed, we did see that each population does have some unique genetic diversity. You can see that here. This is the 14 unique bands, 18 unique bands, and I thought that was interesting that the bigger population had fewer unique bands than the smaller population. Some of the other statistics that she presented, she did a pairwise comparison. And this basically shows that indeed the populations within Joshua Tree are more closely related to each other than they are to the one outside of the park. Interestingly, the Covington Flats population, which is the one that was farther east, was more closely related to the Catclaw Flat population outside. And in fact, the Erigeron work, which I'll show you next, it sort of mimics exactly what we see here. So I'm going to try to fly through that one too, so I just thought I'd bring that up now. It's basically the same story. Also the genetic structure, so we sort of focused on the GST values because it's the most conservative value of the 3. And the value basically tells us that, or suggests that the populations have not greatly differentiated genetically, so there likely is some gene flow, either past or present, occurring. Also, if you use these GST, the values to make some predictions about life history for the plant, the GST values actually, it does fit pretty well to what we know about Astragalus tricarinatus. It's consistent with long-lived perennials, however, so we don't really think this is a long-lived perennial, but that is something that we're hoping to ascertain with our monitoring projects. But it is obviously narrow geographic range. We think it's an outcrossing species. We don't know that for sure. Again, something that we're hoping to learn with time. But if you were to go with the theta 1 value over here, it's a little higher, and that actually falls within an expected range for a selfing species with a wider geographic range. So we're a little more comfortable with this GST value over here. And then there's a number of species that have been studied within the genus, and I guess the genus is sort of notorious for having low genetic diversity. So the 0.164 is actually fairly intermediate when you compare it to all the other studies that have been done in the genus. Here's some just various population statistics and descriptors. The bolding is for high values and the underlining is for low values. So Eureka Peak, again, this is the one with the larger population within Joshua Tree, consistently had the lower scores, whereas the Covington Flat had the higher scores, but really all 3 populations have pretty similar levels of genetic diversity. Not a lot to say there. And then the UPGMA and PCA analysis basically tell us the same thing. Individual populations are generally cohesive, but populations did not form monophyletic, reciprocally monophyletic groups, which supports the idea that there is some gene flow occurring, at least historically, if not presently. And then here's a great picture. I mentioned that the habitat's really steep. So there's a person right here, and she, I don't even know if she's looking for Astragalus. She's probably looking at some little Nemophilus or something, I don't know. But this is where the Astragalus occurs, and you can kind of see the different coloration in the soil.
Microphone, please.
I thought I was close enough, sorry. So so you can kind of see that distinctive color in the soil that I was talking about earlier. So we did attempt to start some long-term monitoring for this species back in 2010. The idea being that we want to establish expected lifespan for individuals to assess longevity and long-term viability of populations, to monitor reproductive capacity, and to establish mortality rates and reproductive capacity for size classes. In 2010, at that time, we didn't know of the bigger populations that we know of now, and so we were trying to access places like this and tag enough individuals to make it worthwhile. And basically, I mean, my field crew came back and they were really mad at me because I, thought that this would work, and they said, no, I don't think we should do this. And they were right. I was out there too, and I think we were doing more damage to the habitat and the species than what it was worth. And so, so we kind of just sat on that for a while until we came up with, we learned more. We have now some populations that are bigger. We feel a little more comfortable about establishing some long-term plots. So now we have, last year we established 2 permanent plots capturing 34 individuals, and they're a little easier to access, so we feel a little bit better about doing that. And just to, so again, these goals are going to be reiterated for the Erigeron. I'm not going to show those objectives again just because it's all the same goals. But with the Erigeron, I was a little more successful with our first attempt. Okay, so Erigeron parishii, this was it was listed as threatened in 1994. It is protected under the West Mojave Plan, and it does have critical habitat designated. It is also a unique-looking plant. In Joshua Tree in particular, there's really nothing you can mistake it for. It has this really pretty gray foliage and the typical light purple flowers with the yellow center. This is, again, it's an endemic to Southern California. This is the global distribution. Occurs in Riverside. This is the county line. So Riverside County, it just barely gets into Riverside County right here in Joshua Tree. Again, this is the park boundary. So you can see that these populations really do represent this, the very southeast corner of the distribution. Also, again, this is the USGS geology map. That's the background. And in this case, you can see that in the western portion of the range, it almost exclusively occurs on limestone or dolomite-derived substrates. But that's really not the case in Joshua Tree. So again, this is the soil map produced for Joshua Tree National Park. And basically, it occurs on granodiorite, gneiss, and Monza granite in Joshua Tree. So it's a very different substrate there. Also with this one, prior to 2006, it was known from 3 historic vouchers. In 2006, we were able to relocate at least what we think represent those 2 occurrences represented by those vouchers. And then since then, we've added 4 occurrences, including significantly here the Quail Mountain population, which is this cluster of points over here that we estimated at around 1,000 individuals. So that's quite a find, considering this is it's a pretty big mountain and it's accessed and visited a lot. So it's kind of amazing that nobody had ever seen it there. We estimate around 208 acres of occupied habitat with about 1,200 individuals in Joshua Tree. So again, we did the habitat model, and in this case, it helped us locate 1 additional population. So we felt pretty good about that, and also it was just using the same script and same parameters as I discussed for the Astragalus. The germination trials for Erigeron showed that indeed this species does not require any special treatment to break dormancy. No treatment yielded 75 to 100% germination rates. Significantly, they had a seed lot that was 25 years old, and they had 100% germination rates from that, so that was kind of fun for them to learn. The genetic analysis, as I mentioned, it's very similar. The 2 populations that were sampled in Joshua Tree, this one we call this is the Quail Mountain population, and that's the big population in Joshua Tree. This is what we call the Long Canyon population. It's a pretty small population, and then we compared it to a fairly large population over in the San Bernardino National Forest. And again, we had unique bands found in each population. and I think I mentioned this already, the pairwise comparison is pretty much mirrors what we had in the Astragalus, where you see the two populations in Joshua Tree are more closely related to one another, but the Quail Mountain population was slightly more closely related to the Monarch Flat population, which is the San Bernardino National Forest one, despite being a little bit further away. In this case, the GST value, again, it supports that the populations have not greatly differentiated, suggesting there is some gene flow, if not now, then historically. It also is consistent with a moderately perennial with limited geographic range, and this matches pretty well to what we know about the species. And in this case, these statistics, the bolding, you can see it kind of wobbles back and forth. So the two large populations, the Quail Mountain population and the Monarch Flat, basically had more similar diversity values than did the smaller population in Long Canyon, which, so that's the underlining, is the low values. And same story with the PCA and the UPGMA analysis. Basically, individual populations are generally cohesive, but populations do not form reciprocally monophyletic groups, suggesting gene flow between populations. Okay, and then in this case, we tagged 52 adult plants and 10 seedlings in 2009. And then we also, in 2013, we established 4 permanent plots for the species that captures 65 plants. So the initial attempt was just to try to get out there and learn a little bit about the species and how best to capture the information that we were after. So some very simple information came from that. You know, we captured the reproductive phenology pretty well between May 1st and May 28th. These are just really pretty graphs to show something very simple. So this is percentage of the plants with heads at a given reproductive stage. These are so going from buds to flowers to seeds. And then this is a median value for total number of heads in each reproductive stage. So again, buds, early flower, late flower, and seeds. And then this is showing that there is a significant positive relationship between total number of flower heads and total plant volume. And the idea behind this is that I'd like to be able to capture some meaningful size classes that help us tell us something about mortality rates, survivorship for the different size classes. So far, the data that we have suggests that until you get to this size class, you probably die, and then you have a better chance of surviving and growing until size class 2 is the 51 to 500 cubic centimeters. And then once you get into the size class 5, which is over 25,000 cubic centimeters, then your chances of dying go way up. But I don't know if that'll really stand up with time, and this is just a first take on it all. So anyway, in conclusion, surveys have been important for a better understanding of the species distribution and abundance in Joshua Tree and range-wide. The populations in Joshua Tree represent the edge of the range for both species, and so I think that it is important for us to be a part of long-term planning for the species recovery. And hopefully the long-term monitoring projects that are being done at Joshua Tree will help address some of the outstanding questions that we have for these species. And if you are interested in looking at the reports, you can email me or find them on the website. And of course, there's a lot of people to thank.